we are back with the second part of our program today. So if you are joining us just now, welcome to the May Institute. We are very happy to have participants interested in the topics that we discuss. And if you haven't seen the functionalities before, so we have at the bottom a functionality chat and another functionality Q&A. So please use Q&A to ask questions to the presenters. And the presenters will either follow up on that, or we will read the questions out loud to them so that they can answer them. And then there's another functionality chat. You can use it just to message the presenters or everybody. But if possible, please use Q&A for your questions because it allows us to monitor the questions a little bit easier. Well, without further ado, let me introduce our next speaker. I'm really excited to have a chance to introduce Brian Searle, who is a translational research fellow at the Institute for Systems Biology in Seattle. So you probably are familiar with the company Proteome Software, which is in charge of the software called Scaffold. So Brian is a co-founder of Proteome Software. He started the company in 2004 and was playing a leading role in the company for almost 10 years. But 10 years after founding the company, he decided to go back to academia. And in 2014, he joined the lab of Mike McCoss in University of Washington, where he developed methods to quantify proteins and especially phosphosides using mass spectrometry. And Brian made really important contributions in DIA. And so since 2 years ago, he joined the Institute for Systems Biology as an independent fellow, and he continues working on proteomics, mass spectrometry, and bioinformatics. And we are very, very fortunate to have Brian as a speaker at the May Institute. We are very grateful that you could contribute your time, Brian. Thank you very much.
Thanks, Olga. Is this working well? Okay, great. So thanks, Olga. That was an excellent session by Lindsay, and I hope you guys all got to see it. And we're going to essentially pick up where she left off, and start thinking about DIA, but from a PRM sort of perspective. So just before we start, I'd like to just say that I'm going to do my best to answer our questions as they come up, but I'm probably not going to do quite as good of a job as Lindsay did. If I don't get to your question, then please either send me a message on Twitter or send me an email at this email address. Okay. So the ISB requires that I do this speaker disclosure just to start before I give presentations, just to say that I'm a fellow at the ISB, and then I also am a founder of Proteome Software as well. So with that, I'd like to start to get into one of my favorite topics, which is what would you do if you were interested in finding, say, for example, my favorite book in the library, say Harry Potter and the Prisoner of Azkaban, and I might want to go and try to find this book. You know, if I were going to the library to do this, what I probably wouldn't do in this would be to take a bunch of books off of the shelf, maybe take the biggest ones first, and then pull them out on a cart and then go through them one by one and say, well, was there Moby Dick there? Or what is present in this library so far? Is Moby Dick here? Is Pride and Prejudice here? War and Peace here? I didn't find Harry Potter when I pulled these books at random. That would be kind of a ludicrous way to navigate a library, right? The correct way would be to use some prior information about those books, know that they're present and written by J.K. Rowling, and just go to the R section in the library and then pull the book off the shelf, or know that it's definitely not there. Okay, so obviously this is kind of hyperbole relative to DDA, but I just sort of like to start with this notion that failing to detect a peptide is not actually a proxy for its presence or abundance. So just to kind of go through some of the basic schematics for how DDA is, just to make sure that everyone's on the same page. With DDA, you would be interested in identifying peptides out of a precursor map. So over precursor MZ on one axis and retention time on the other, each one of these peaks is a precursor signal for a different peptide. And the way you would typically do this with DDA is you would pick at a given time point in retention time, you would pick a peak to go and collect an MS2, a sequencing spectrum essentially, and then use that to determine what the sequence of that peptide was, and integrate the precursor peak to say how much of it was actually present. So you would pick that point, you would stick it on an exclusion list, and then you would go down to the next time point, and then you would pick the next most abundant peptide, go with that, sequence it, and then go to the next one, go to the next one, go to the next one, go to the next one. Now, not every peptide is going to be one of the most abundant peaks at any given time point. And so not every peptide is going to get sequenced. In addition, all you have to indicate that this peptide is actually present is a single spectrum for it. So just to contrast this to the PRM example that Lindsay so beautifully explored, if you were to think of this same precursor space with PRM, you would specifically select peptides that you were interested in targeting and pick their masses, and then scan through no matter whether you saw precursor signal or not, and always collect a fragment scan for those peptides. And so one of the things that's interesting here is that you get these quantitative fragment ion peaks, as Lindsay so clearly showed earlier. Okay, so why is this important? And I think one of the things that's sort of hidden under the covers with DDA is really the instruments only have so much time before they can start to go through and have to work on the next batch. So for example, many methods are top 12 or top 20, and they'll go try to sequence these top 12 most intense precursors before having to look at the next spectrum or the next precursor scan to derive more sequencing spectra. And this is really important for a variety of reasons. Let me show you, for example, here. This is about 60 seconds of peptides as they're eluting off the mass spectrometer. So each one of these scans is a precursor scan, tiled one after another, so you can see them essentially in real time. And you can see peaks rise and fall all of the time. And so every time there's a precursor scan, that's a new batch of peptides to try to sequence. And so there's going to be regions of this proteome that are just never going to get above the sort of limited threshold of being able to be one of the top 12 peaks. And so this is what's one of the reasons that it's really important for dynamic exclusion to work. But it also indicates that there are peptides that are just never going to be sequenced, even though they show really clean rising and falling like a peptide should behave. So just in that one batch, I just took this 60 seconds of peptides and highlighted every precursor that had a total integration above 1E7. So that's, and this is on a Q exactive. So 1E7 is a very high intensity. That's a very confident intensity that you would trust. And if I sort of flip that on its side, I'm just going to show you all of those precursor peaks. There are actually, in this 60 seconds, there are more precursor peaks in this 60 seconds than you can collect MS2s on that same Q exactive HF instrument. And it's just indicates just how rich this space is of peptides that we're interested in trying to monitor. Let me show this a little bit differently. So in this particular plot, I've got retention time on this axis, the x-axis, and I've got on the top plot, I've got the tick intensity, so the total intensity. And so you can see that rise and fall, starting to rise around 20 minutes and then peak at around 50 minutes and then sort of tail off all the way out to 90 minutes or 100 minutes. And so on this bottom axis, or this bottom plot, I've got the number of MS spectra that were collected. And you can see that it peaks out very quickly, right? So the instrument is collecting MS scans as fast as possible, starting at 25 minutes, and it continues to do that all the way out to 110 minutes. And in green, you can see what are the number of PSMs that were detected, the number of peptides that were detected at any given time point in that region. And one of the things that's really interesting is that in this region where you actually have fewer peptides, right? There's less precursor tick signal. You actually end up detecting more PSMs or more peptides with DDA. And the reason for that is because you have much less complexity in terms of co-eluting peaks or MS that actually contain a single peptide at a time. So I think what I'm trying to underline here is that the spectra that we're acquiring, even when we're looking with DDA, are already multiplex. They're already very complex, and that we're actually only able to achieve our highest detection rate in this region where there are fewer peptides than we actually have in the middle part of our gradient. Okay. So PRM is really great for being able to detect these both difficult to identify peptides because they're so low in intensity, but also very good at selecting out specific peptides for measurement. But what if you wanted to do this across an entire genome, and is it possible to target every peptide? And it is with DIA, but there are definitely some compromises that you have to deal with. So this will get to the sort of contents of the talk that I'll present here. One is to sort of discuss all of these compromises that exist in DIA. So but thinking about it from a global perspective for PRM. And then try to talk about a bit about how to analyze DIA data, not specifically from any given program, but from DIA data analysis software in general. I'm going to discuss some of the pitfalls that you might see with looking at DIA data and what makes it difficult, but then also talk about how to set up a DIA experiment, as well as how do you build libraries specifically from a DIA-only data set. Okay, so what does DIA look like from the framework that we have laid out here? So again, if we were to look at this same precursor map, instead of selecting a specific precursor, a specific peptide to fragment, we actually select a range of peptides to fragment simultaneously. And so we'll scan across for those time points and collect series of fragment ion spectra that are multiplex. They contain peak information from multiple peptides at the same time. So if you look at any one of these scans, they're extremely complex. They'll be representative of all of the different peptides simultaneously. But it does give us a chance to at least be able to do PRM style consideration if you look at coelution of these particular peaks. So all of the purple peaks coelute, they line up with each other. And so because you can look at how well they line up, you can say that that represents a possible peptide. Okay, so we can cover the entire proteome, but again, at a cost, right? There's this increased complexity from the multiplexing. So how are we trying to make these types of measurements? And really, the compromises that you deal with this type of acquisition are set out by the constraints that you have to work with. So the instruments that we have are only able to scan so fast without being able to make measurements, or with still being able to make measurements. And usually the type of instruments that we're talking about might be able to go from 10 to 20 hertz, so 10 to 20 scans per second, as much as potentially 40 scans or 50 scans per second on some QTOFs. And the other constraint is that to describe peaks, as Lindsay showed, you really need at least 6 to 8 measurements across the peak to be able to adequately describe its quantitative level. And so for this, there are several compromises that you need to consider making with DIA. One is that, as I said earlier, you have to accept a higher level of interference in these windows by looking at co-fragmentation of multiple peptides at any given time point. Each of the MSMS scans typically are actually, you typically will spend less time with DIA parking on each MSMS scan than you would with an equivalent PRM experiment. And then secondly, or thirdly, that you end up typically needing to limit the measurement area that you're interested in considering. So you typically can't scan the entire DDA range, which most people would acquire from, say, 400 to 1600. Typically, we have to look at a more limited range from 400 to 1200 or 400 to 1000. Okay, but this data, if you were to look at it from the Skyline perspective, or just from the fragment ions that are presented, if you were to look at a specific peptide that was shown in a PRM experiment, you would actually see a really similar signal for that with DIA, except there are these sort of confounding issues with these constraints. One is that you're going to get a lot more interference because there are a lot of multiple precursors at the same time. So for example, this big purple peak over on the left of the DIA peak is an interference. You have fewer points across the peak, typically, because you just can't, you need to spend a lot more time scanning lots of different windows. So your ability to trace across is maybe not typically as good as with PRM. And so this is where that constraint of 6 to 8 points across the peak is really important. But that said, if you were to look at this, the actual intensities that we're measuring, and this is just a PRM on a QE and DIA on the same QE, they actually present very similar types of data with very similar types of measurements, just with slightly lower fidelity with DIA. Okay, so when we think about a normal DIA experiment, okay, so there's actually a couple of different questions. I'm going to start with these questions to go. One question is there any difference between DIA and swath? Swath is essentially the SciEx brand name for DIA. There's actually not very much difference at all. It's just a specific way of acquiring DIA. DIA is a more general term that spans across instruments. The second one is how good is the quantitation in DIA? We'll get to that in a bit. Okay. So thinking about a typical DIA experiment, we would acquire a precursor scan, say, for example, between 400 and 1000, and we'll acquire several fragment scans, so one after another, until we'll scan through an entire range, and then we might acquire another precursor scan, and then again, another set of fragment ion scans after another. Always the same window, one after another, between each cycle. So in this case, I've represented 1 and basically 1.5 cycles here. Okay, and so you need the time window between here, which will essentially indicate the number of points across the peak. So for example, if you had a 25-second peak and your cycle time here was 2.5 seconds, then that would give you essentially 2 points across the peak. Okay, so I'm going to take that entire fragment ion scan and then sort of smush it up so that we can see it all together. And then I'm going to take a look at one individual fragment ion scan. So this one is from 784 to 808, encapsulating one particular peptide that I'm interested in making a measurement of. If you look at that scan and it's of a fairly intense peak, as this one was, then you would actually see really nice fragment ion scans, and it would look sort of like a DDA measurement. But we actually acquire these one after another, and so we actually get these nice sequential scans, again, one after another. And there will be some noise associated with that. But if you look at it down this one axis, again, it'll look like PRM data in that it's that you get to see chromatograms. Okay, so there are a bunch of caveats here with this type of experiment. And I think that it's important to go through these caveats first before answering questions. So for example, if we're trying to measure a specific peak or a specific peptide in that window, I think it's important to remember that ions are not necessarily centered on a specific or the MSMS are not centered on the specific ion of interest, and it's not necessarily collision energy optimized. So for example, if this is a +2 peak and you were acquiring spectra assuming that all peptides were +3 peaks, then it might not necessarily be the right collision energy. Another thing is that, again, as we said, there are usually multiple peptides that are simultaneously co-fragmented in each one of these windows. Luckily, we get to see some of them rise and fall over and over again, and you can use the information about that rise and fall to determine which fragment ions go with which peak. And then the second aspect to this is that some of these peptides are going to exist on the boundaries of the window. So for example, this red ion peak, and these peptides will actually have decreased transmission efficiency in the quadrupole. So they won't fly or they won't be transmitted into the orbit trap quite as well or into the instrument because of the sort of window boundaries. And so typically we think of this as a slightly tapered transmission. Okay, and so before we get into what does data look like, DIA data look like, I'll try to answer a couple of these peaks or a couple of these questions. So when considering peak widths, when we're thinking about, typically when I think about peak widths, I think about the base to the base, right? So rather than the actual half height, because some types of peptides will be very large or tend to be acquired across a large retention time and have very long tails. So typically, even that level of information is actually still useful for helping to describe the shape of the peak. So if you still have long tails, that can actually be, in some ways, not necessarily helpful, but in this case, still appropriate for describing the shape of the peak. So how unbiased is DIA? I think that DIA is biased by how you set up the type of experiment. So for example, I just mentioned that you have to fragment all of the peptides as if they're one specific charge state. Because each MSMS scan can only be acquired for one charge state. So for example, if you set that default charge state to be +2, then what's going to happen is all of the +3s are going to score less well than the +2 peptide. So there's already implicit bias that is part of this type of experiment. Now, that said, I think it is less biased than DDA. For calculating the cycle time, in this particular case, it's just the time in between repeated measurements. And so you can see, basically looking at a raw file, you can look at what is the average distance between the same scan from one cycle to the next, and then that will vary a little bit, but it won't vary a huge amount across the experiment. So that's typically a good way to start to think of this. And the next question is about overlapping fragment scans. We'll get to that a little bit later. So that will be definitely discussed. And then as well as what sort of how we actually set up the instrument. Those are the types of things that we'll definitely be discussing later. So we'll get to those as we go. So before we actually start to get into how to acquire DIA data, let's just talk about what it even looks like to begin with. So for example, this is a particular peptide from a protein superoxide dismutase that I was interested in studying in human cells, and this is in a HeLa experiment. And so if I look at all of the extracted fragment ions across all of my retention time, there are a lot of peaks. We see a lot of signal for these different fragment ions that could be generated by this peptide. But if we zoom into this one specific region where we expect to see this peptide, then we actually see a really nice signal for it, just like how Lindsay was showing with PRMs. This is a beautiful peak that you would easily be able to integrate across and be very confident in the type of measurement that you're making. Okay, so in this exact same experiment, I had this other peptide, also from superoxide dismutase. Again, these peptides should be roughly equimolar, although not necessarily. And again, we see the same sort of signature of lots of, or the same or fragment ions from this particular peptide strewn across all of this retention time space. Okay, so if I were to indicate the actual location of the peptide, this is where it would be. And if I zoom into that region, there's a lot of different possibilities for what that peptide could be. Is it the peak on the left? Is it the peak on the right? Is it that peak in the middle? And it's typically pretty difficult to make decisions as to which is the right peak to actually use for the integration. So in this particular case, it's this peak on the right. And I want to show you how I came to that decision. The way I can do that is I can look at my integrated peak area across the retention time space for that peak on the bottom. These would be the intensities of all of the different fragment ions that I was measuring, and then compare that to a library spectrum up above that shows a lot of the same kind of characteristics: a high Y8, a high Y9, a nice ladder from Y10, Y11, Y12 all the way through. That Y2 is also intense, but it's not a very selective ion. And Lindsay actually mentioned this as well, that that some of these lower mass ions are not necessarily as indicative of one particular peptide or another. But the data that we're looking at is remarkably noisy. So this is the MS2 at the actual apex of that spectrum. And so it can actually be pretty difficult to pick out Y8, Y9, Y10, Y11, Y12 for this particular peptide and feel confident about that measurement. And the only way that we can really do this is by thinking about retention time or these types of measurements over time, because any individual measurement doesn't have a lot of fidelity. So one way to sort of look at that is here. If I actually pick out the fragment ions that seem like they coalute versus the fragment ions that don't seem like they coalute, the green ions actually line up really well, while the yellow ions don't line up so well, don't quite line up. For example, you could use them for detection purposes, but you wouldn't necessarily trust them from an identification perspective or for a quantitation perspective. And the red ions don't even seem to agree with the shape of the peak at all. These are most likely just caused by interference from other types of peptides. So really, I think it's important to hammer home that some of these peptides actually look beautiful, like PRM data, but many of these peptides look like this. They look kind of crummy, where you have to make decisions based off of some of these fragment ions where coelution is really much more important than the actual intensity of those in any individual MS2. So you can't really think of this from the same perspective as DIA or as DDA. Okay, so why would you want to use DIA if it is so hard to interpret? And I think that it really comes down to this sort of issue. So if you're thinking about doing an experiment with a PRM focus or an SRM focus, first, you would have to come up with a hypothesis that you wanted to test with your experiment. You would have to select some peptides, as Lindsay described, from the proteins that you would be interested in measuring. You'd have to buy those peptides or make those peptides, and then figure out what sort of retention time scheduling that you would need to do for those. You would have to pick transitions that you trusted for quantitation, and then you could actually start doing data acquisition. And then once you acquired that data, you might actually come back and say, well, did my hypothesis make sense? Should I actually have selected different types of peptides or different types of proteins even for making these types of measurements for the study that I'm interested in? I want to contrast that workflow to DIA, where the first thing that you do is you actually acquire data. So rather than having to iterate through this process of coming with a hypothesis, selecting peptides, acquiring data, then selecting new peptides, and then acquiring more data, Here we only acquire one data set. And then afterwards, we can generate a hypothesis about what peptides we want to extract out of that DIA data set, and then refine transitions using automatic transition refinement or software-based methods, test our hypotheses, and then decide if we needed to extract other types of peptides or other types of proteins from that same data set. So again, you won't necessarily need quite as much sample in these particular cases. Okay, so this is a good point to sort of back out before we start talking about analyzing DIA data, and I'll answer a couple of questions. So is it more beneficial to increase depth of coverage and quantitation accuracy or scan speed or increase resolution? For this particular question, I think that really depends on the type of experiment that you're doing and how many peptides you feel like you need to acquire across a range. So oftentimes experiments, I feel like, do better if you use a narrower precursor range and you have very good isolation. But that may change depending on the type of experiment that you're trying to do and what are the limitations. Again, we'll get into some of the details for why you would want to make certain selections later down the road. So there's another question, DIA versus DDA. In this particular case, I would vote for DIA. For these types of experiments, if I am interested in doing a detection experiment where I've only got like a single sample or a handful of samples, and I just want to do a spot check, I'll typically do a DDA experiment. But if I want to do a quantitative experiment, I will much more prefer to do a DIA experiment than a DDA experiment, unless the sample has very low complexity and the things I'm interested in measuring are relatively high intensity in that matrix. Okay. What is the effect of the taper on the relative quantitation of peptides and proteins? So because it's the same windowing scheme is actually going to be used in each sample, so in each acquisition, you're going to use the same windowing scheme. And so the same tapering effects will be consistent for that peptide in each one of your experiments. So relative quantitation is going to be fine, but you'll actually see a decreased overall intensity in that peptide than you would otherwise be if you had targeted specifically for that peptide. So there's another question about using staggered windows. Again, we're going to talk about staggered windows a bit in a little bit later. So the advantages of library-free versus library-based, we'll also talk about that one later too. And then why don't we use MS1 for doing our quantitation? And I would argue that there are some reasons why you might want to use MS1 if, again, your sample is of low complexity and you actually don't see that many precursors simultaneously. Then I think using MS1 quantitation is great. There's no problem with that. In those cases, you probably just wanted to have collected a DDA experiment in the first place. DIA excels when the matrix is very complex or the things that you're interested in are very low abundance. And the reason why is because when you're co-fragmenting peptides in a small range, so for example, from 500 to 525, you only have to worry about peptides in that same range. Whereas if you're looking at a precursor scan, you're having to make the measurement of your 500 to 525 range, but also have those signals fight with other signals elsewhere in the precursor space. So again, this is one of the reasons why Boxcar works fairly well, is because it actually has, it takes that precursor space and divvies it up into smaller segments where you actually can fill specifically for those smaller mass ranges. But one of the reasons why I think precursor intensity integration isn't as sensitive typically is because of that competition between the ion that you're interested in and other more abundant ions that are being acquired at the same time, especially when you start to think about only being able to use, like, for example, an orbit trap, where you can only put in 1 million charges into the orbit trap. before you end up with space charging effects. These types of issues make it much more complicated to be able to fill with the ions that you're actually interested in measuring. So this seems like a good spot to go back to thinking about analyzing DIA. And so thinking about how we make our measurements, the part that you have the most control over is actually the peptide selection. And as part of the analysis procedure. And so there are lots of different ways that you could think about peptide selection. And again, this is whether you're using a library base, for example, or whether you're thinking about all peptides at the same time. So there would be, you could look at, for example, the most broad analysis, which would be looking at all tryptic peptides, all the way down to only looking at peptides that are acquired by DDA from your experiments in auxiliary runs or library-based runs for those specific experiments. And so Lindsay put up this slide showing specifically Cas-Z1, which is a transcription factor, and just the transition response for different peptides in Cas-Z1 sorted in order, and how in this particular case, there's 3 orders of magnitude or more than 3 orders of magnitude between the top responder and the bottom responder. This is actually really common. This is pretty normal. So if I look at, for example, this is a sampling of 700 different proteins, again, all synthesized proteins. So we know that their peptides should be at equimolar levels. This is the orders of magnitude between the top responder and the bottom responder. And on average, it's about 3 to 4 different orders of magnitude for each individual protein. So trying to monitor every possible peptide for a protein is just an extremely difficult problem, and I think is essentially a red herring of the way we typically think of analyzing DDA data, where it's fairly easy to look across all particular peptides for matches for a single spectrum. And we'll get to why that is the case a little bit later. But most of the peptides that you'd be interested in monitoring actually don't produce a very good signal at all. So, and potentially don't even produce any signal that is actually monitorable in your experiment. So typically, we think of trying to analyze these types of data sets using spectral libraries or using chromatogram libraries as sort of the better way to sort of approach these types of experiments. And as I said earlier, we'll typically take a library of MSMS acquired from DDA, and again, this is a particular peptide that we had seen in a library, and then we would try to make those measurements by matching those particular fragment ion patterns and which fragment ions are present to the DIA spectrum. Typically, these libraries contain a target mass, so the mass of the peptide, the relative intensities of the fragment ions, the retention time for that particular peak, and sometimes they'll contain some information about peak shape or what is the likelihood of a peak being interfered with in the case of chromatogram libraries or DIA-based libraries. Okay, so what are the advantages of doing a spectrum library analysis? And I think there are 3 really important advantages. One is that you know something about fragmentation. And I would argue that you only sort of know something about fragmentation, actually. So for example, if we consider this experiment from the Kuster lab, where they took the same peptides and analyzed them on a Lumos in-house and then on a Q-exactive in-house for example, in this top plot at a fixed NCE of 32, the fragmentation patterns are actually completely different for these two, even though this is a spectrum that is acquired on the same vendor's instrument. And again, this is a butterfly plot showing the top versus the bottom. And so actually, one of the things that they found that was pretty interesting was that there's actually kind of an interesting spread of about maybe 6 NCE between these 2 classes of instruments, between the Lumos and the Q-exactive, that these normalized collision energies don't necessarily mean the same thing from one instrument to another. They actually found something that was sort of mind-blowing to me, which is that even on the same a model of instrument, even amongst two different lumoses, they get slightly different fragmentation at the same fixed NCE, so that each instrument essentially needs to get tuned, or the fragmentation patterns are specific essentially for each instrument. So here I put in some rough conversions so a lumos using an NCE of 33 is roughly equivalent to a Q exactive with a normalized collision energy of 27. But these are, again, rough guides for these types of measurements. We also know something about the retention times for these peptides, but oftentimes in these large libraries, we see that there's a lot of scatter between one type of measurement and another. So from our DIA measurements versus our DDA measurements. So we'll know that it's within a band. So in this case, the blue band, we would say that this is the right retention time window for this particular peptide, but we typically don't know it exactly. And then I think the most important factor is actually having a relatively narrow list of peptides. And so, for example, if I show this on the bottom, these are 4 different sort of search spaces that you would have to consider if you were interested in trying to monitor either just the peptides in a HeLa spectrum library, which is around 100,000 or 120,000 different unique peptide sequences, versus the entire size of the human FASTA database with no miscleavages is already around 1 million different peptides that you'd have to consider. And if you start to add in different potential post-translational modifications like phosphorylation, that number skyrockets, even though we're not considering any miscleavages or any unusual charge states. Okay, so how do we interpret this data? I showed these plots earlier where you extract out all of the fragment ions across all of retention time. And the way we actually end up doing that is by, at each different time point, scoring how well our library spectrum matches to the acquired intensities at the fragment ion level. And so typically we would hope that there might be a peak at some point, and at that peak, that would be the best indicator for that peptide being present. And so this is how we typically think of peptide-centric analysis, where we have a specific peptide that we say extract out all the fragment ions, score all of the fragment ions across time, and then pick the best peak. And so all of the measurements are based off of a peptide-based measurement instead of off of a spectrum, because you take that peptide and compare it to all spectra, as opposed to a spectrum and comparing it to all peptides that are appropriate. Again, typically we analyze this data using the same types of methods as we do with DDA, with the concept of target and decoy. So we will typically use a forward database and a reverse database, but rather than a FASTA database, we'll actually use a forward spectrum library and a reverse spectrum library. So how do you create a reverse spectrum library? Typically, and this is how it works in Skyline and how it works in Encyclopedia, these software tools, is where you actually take the sequence and you reverse the middle part of it. So you keep the termini, or the N-terminus amino acid and the C-terminus amino acid. You reverse everything else, and you keep all of the ion intensities. So Y5 is always the same intensity as Y5 is. And you basically just move the masses and potentially the mass errors that you would see. And then this presents a relatively reasonable decoy for being able to detect peptides. It is not necessarily the best decoy, because we do know that this scrambled peptide sequence would actually fragment slightly differently than we've laid it out here. So there may be potential biases in terms of how we think of target decoy, but this is a reasonable approach, I think, given those sort of constraints. And so a lot of you guys are familiar with spectrum-centric searching, which is what we typically do with DDA. And I just want to contrast what we're doing here with that. So with spectrum-centric searching, you might have 30,000 or 50,000 MSMS spectra that you're interested in making measurements. And you would say, okay, for each one of these spectra, what are the possible peptides that we see in a protein sequence database? And each peptide spectrum match, and basically our currency is a peptide spectrum match where we have one PSM per spectrum that we acquired. So we've essentially asked 30 or 50,000 different questions and come up with 30 or 50,000 different answers. So I want to contrast this with peptide-centric searching, where we've got a list of different peptides. So for example, if we took that entire FASTA database and we were looking at 1 to 2 million peptides of interest, we would extract out each one of those, and then we would have an evidence for that peptide being detected in that sample. Now, this is actually a really great way to think of it, because we actually have, for every different peptide, whether we think it's actually present above a detectable limit or not. And so we have that measurement for every possible peptide across the genome. But at the same time, we've also asked a lot more questions that we would typically ask in a normal DDA experiment. And because of this, we end up having a much more complicated false discovery analysis question, where we have to correct our possibility of seeing false discoveries against having asked way more questions than we would typically ask. Okay, so this is a good point to take a minute and answer some questions. So one of them is, how many transitions do you require for identification? Typically, that actually depends on how you set up an experiment. Most of these types of experiments where you use automated software, again, like with Encyclopedia or Spectronaut or Scaffold DIA or something like that, you actually have a minimum number of transitions, say maybe 3 transitions, but then it's on a peptide by peptide basis for whether that is actually a good match or not. So another question, can using a small library bias your results in some way? Yes, using a small library actually ends up having you ask fewer questions, but at the same point makes it so that you end up in a situation where you might match fragment ions to the wrong peptide sequence or the wrong spectrum in your match. And I want to show you an example of that, but again, a little bit later. So we're going to talk about retention time just in a minute. And so if you're interested in defining a proteome for a specific type of tissue or something like that, there is an advantage to using DDA for some of these types of exploratory questions, where all you care about is what are the possible peptides that are present in a sample. If you're interested in doing a quantitative experiment, though, I would argue that DIA would be a better measurement. But if you're just interested in detection and just having a long laundry list of all the things that are possibly present, then that would be a good way to go. DDA would be a good way to go. And in terms of bias, in terms of hypothesis generation, that actually is kind of attributed to this question of library size. So for example, if you use a very small library, then you end up potentially biasing your ability to detect peptides. Obviously, you can't detect peptides that are outside of that library, for example. Okay, great. I think that let's see. So if you have a sample of unknown complexity, I think that there, again, the real consideration here is whether you're interested in generating a laundry list or doing a quantitative experiment. And that is sort of the question that I would focus on, less about the complexity. I think most data sets are more complex than we think they are. So for example, or the things you're interested in measuring out of them are much lower abundance. So in some types of situations where you have a very high abundant protein and that's what you actually care about, then I would argue that DDA is probably just as fine if you have a low complexity experiment for quantitation. But in other circumstances, I think that for DIA is typically a better method for quantitation. Okay, and we're going to get to things like library choices as well as in silico libraries in a little bit. So I want to first talk a little bit about retention times, because one of the sort of things that's difficult about this type of work that we've got here, and one of these issues about asking so many different questions, is that we actually need to be really confident in the types of peptides we're making measurements of. So we need to use a lot more information in defining peptides than we had before access to. So typically with DDA, it's totally fine just to use a sequence spectrum model to be able to be confident in the peptide detections or whatever search engine that you're interested in. And they don't have to know anything about a library or retention time or anything like that. They typically do really well in those circumstances. But because we're asking so many more questions, we need to actually use or leverage a lot more of this information. One of these pieces of information is retention time. So one of the sort of best ways to kind of deal with this is to use something that was developed called IRT peptides, or index retention times for peptides. And so this was originally proposed by some people in the biognosis group, where they were interested in taking a bunch of these different standard peptides that you would buy from them, and then using those, attributing a specific retention time profile for them, or one of these IRTs here, that is a dimensionless retention time. Essentially, it has no meaning in terms of seconds or minutes or anything like that, and just sort of stratify them. You always see, for example, peptide A before peptide B before peptide C before peptide D if you use C18 in this particular scale. So this is, I think, a really great way to try to deal with this. And if you have peptides like this, then that's fantastic. I think one thing that's important to note is that that any peptide can be actually function as an IRT peptide, as long as you have a way to put it on an index retention time scale, right? In terms of a dimensionless scale. The actual dimension itself isn't that important, nor are the peptides themselves. So for example, you can buy peptides from Biognosis, but you can also buy peptides from anywhere you want. So for example, if you typically use Pierce peptides, which is typically what I use for these types of experiments because they're nice and cheap, then those will be what I would use for my sort of study. Alternatively, you can buy a bunch of peptides, or you can use digested peptides from BSA, for example, which are pretty easy to come by. And so the way you would construct a library with these stable isotope labeled peptides, again, IRT peptides or any other types of peptides, is that you would have a handful of these peptides put into a sample that you would be interested in trying to measure. You would identify using DDA all of the sort of retention time points for all the peptides you were interested in monitoring, and then line those guys up on your scale. And now you have essentially IRTs for all of those different peptides. And you can use that to compare against another DDA run where, again, you injected the same types of peptides into that same experiment. Whether they're IRT, officially IRT peptides or not, it doesn't really matter as long as they're on the scale. So this is one way to generate these sort of larger libraries. Another way is something that that Rob and I explored in this phosphoproteomic experiment. And I think this is an important sort of thing to work through because it actually enables you to start to think about building libraries from data that you already have. So we had acquired a whole bunch of data already. And in this particular case, we have a particular sample that we were interested in monitoring. So this was a phosphopeptide sample that we stuck these IRT peptides into here. And again, these were the Pierce peptides. We did this alignment just like before. And what we ended up doing was deriving IRTs for all of the peptides we were able to identify. So 5,000 peptides in this particular case. Now we use those 5,000 peptides now because they now had IRT values. We could use those 5,000 peptides to map to all of our previous data sets. So for example, we would be able to map them to this particular experiment because these are endogenous peptides now that we have IRTs for, and we can put them on the same scale. And then you could do that for all of the different types of measurements that you've got. And so we ended up collecting this enormous library of phosphopeptides that we published over 1,000 different runs here from different labs in this particular case. Of IRT peptides or IRT phosphos standards now for our experiment. And so these are, again, all endogenous peptides that we didn't have to buy or synthesize. We could just do using this type of method where we had one sample that had IRT peptides in it at one point, and then we can map all of the other samples to that. Okay, so there's a bunch of these libraries that exist already. Some of them have very good retention time characteristics. Some of them, like the NIST dataset, don't have retention time in them. But these are a list of publicly available libraries that I encourage you to go and look at. Because I think that this is a very good source for taking these measurements. I'm not going to talk about any specific tools here, but there are lots of different tools that can use either these library methods or try to analyze DIA data in general. And I just want to point out that these tools exist in essentially 3 different analysis frameworks. One framework is to use a retention time filtering based method, where you have a retention time in a spectrum library and an IRT in a spectrum library, and you want to only interpret peptides that fall within a range around that peptide. And that's a kind of a nice way to eliminate all of the possible false positives that you might actually see. So there are a bunch of these different programs. Many of them are based on the mProfit model, which is, again, built into Skyline as well. So I want to contrast this with tools that are retention time agnostic. So for example, there are some tools like Pecan and MS Split that don't actually consider retention time at all, or tools like Encyclopedia or Scaffold DIA that use a first pass analysis to kind of judge how confident that you are in any different retention time, and actually function in situations where you don't actually have retention time measurements whatsoever, that are designed essentially to look for every single peptide across the entire retention time window. And then contrast this with tools that are designed to actually use the original sort of DDA-like platform for analyzing data using search engines. Tools like DI Umpire or PLGS from Waters are great sort of ways of interpreting DIA data as well. But essentially trying to convert DIA data into DDA-like data to be able to process it through search engines, normal search engines that you would have for your DDA platform. So I want to kind of finish this small segment before getting into the next bit with this idea about how accurate are these types of measurements from a quantitative perspective, because this is something that's been raised a few times. And so here, actually, I'm showing an experiment where I've did a bunch of PRM measurements. I made 500 different PRM measurements of a specific data set in which I had peptides that were, many of them were changing. Most of these peptides that I selected for the PRM were changing. And then I compared to those to DDA and DIA, just in terms of the number of peptides we could detect, as well as how accurately did those types of measurements recapitulate what we saw with PRM. And so one of the things that we were looking for here is that if you look for the ratio of ratios, you would hope that you would get a ratio of ratio where the DIA data set agreed with the PRM data set, or the DDA data set agreed with the PRM data set. And that would be essentially a 0 in these cases of a ratio of ratios on the log scale. So one of the things that you can see is that all of these measurements actually hit that 0 reasonably accurately, no matter whether you use a normal DIA experiment, this is just 25, 24 MZ wide windows, or variable width windows, or using staggered windows. They all hit about that same measurement, and they all have about the same accuracy in terms of fidelity. The only difference is really just the number of types of peptides that you can detect based on having either a variable width region or these staggered regions where you actually have essentially narrowed precursor isolation windows. The width of these boxes, or these errors around that 0 point, are typically more narrow than DDA. So these are box plots where the box itself represents the sort of median set, the interquartile range, essentially the middle 50% of the data set. So if we start to think about this as if you were to take that box and expand it out, making it larger, 60%, 70%, one of the things that you'll see is that the DDA measurements actually get really noisy quite quickly. And the accuracy of the DDA measurements in these particular cases, you might trust about 70% of those measurements, but another 30% of those measurements are maybe not actually accurate at all in terms of their actual ratios of intensity between one experiment and another experiment. And I want to contrast this with the DIA data sets, which all for the most part performed about equally, no matter whether you use normal DIA or overlap DIA or staggered DIA or variable with window DIA. They all performed pretty admirably out to about 95% of the data set, and that only in the last 5% that we would see these errors creep in. And so this is actually really important, I think, that one of the problems we typically have with DDA is that we make detections and then quantify everything and say they're all good measurements now. And I think one of the issues that we have with precursor ion integration is that actually not all of those measurements are actually representative of reality. Many of them actually show interference. But when you only have a precursor, a single peak or a single set of peaks to make a quantitative assessment of, then you don't actually have very much information in terms of you have no ability to throw out particular fragment ions or specific measurements. But with these fragment ion profiles, you can actually just start tossing out fragment ions that don't agree with the rest of the peaks based off of the peak shape. And so it's much easier to be able to decide, do these measurements actually make sense from the standpoint of whether they essentially agree with each other. And because with DDA, you only start with maybe 3 measurements per peptide per sample, so a monoisotopic, a +1, a +2. With DIA, you typically have many more measurements, and you can actually delete down lots of measurements. And so oftentimes you have, so in this particular case, 80% of measurements have good transitions, and these are transitions that follow the shape of the peak. About 80% of the time you get at least 3 of them as being good measurements. And because of that, you actually get really good coefficient of variance for these types of measurements. And I think this is the important reason why DIA succeeds, is actually it's because you have the ability to throw out data, not that, or you have the ability to decide if your measurement is accurate and then throw out data as you need to get down to the tightest types of peptides or the tightest types of fragment ions that represent that peptide. Okay, so I will talk about potential pitfalls, but I'm going to answer a couple of these questions first. I'm going to start actually with the bottom at this point. So okay, so in terms of the difference between peptide-centric and spectrum-centric approaches, it's very subtle in terms of the explanation, but it actually is a pretty fundamental difference in how you try to think of these experiments. And so if you have a spectrum-centric approach, then you are taking a selection of spectra, and you say, okay, for each one of these spectra, each one of these can be at most 1 peptide. So then we try to assign for each one of the spectra a particular peptide as a PSM. So the number of times we ask a question is actually limited based on the number of spectra that we've got. So if we've got 50,000 spectra, then we only ask 50,000 questions in that particular case. In a peptide-centric mode, you actually take your entire laundry list of all possible peptides, and then you ask them one at a time, is there any evidence for this particular peptide in my data set? And because of that, you have a lot more questions you have to sort of ask, essentially, and then justify based on false discovery. But you also have the ability to answer, is this peptide actually not present in my sample? And so that, I think, is the big benefit of this approach. But the big negative of this approach is having to ask a whole bunch of questions. Now, a lot of people think that you can only ask spectrum-centric questions of DDA and peptide-centric questions of DIA, and that's not true at all, actually. So, so as I said earlier, DIA umpire and PLGS from Waters try to take DIA data and convert it into a framework that you can ask spectrum-centric questions about that data. Conversely, other people, so Bing Zhang, for example, has developed a peptide-centric search engine for DIA data, for DDA data, that can scan for any particular peptide. Do I have evidence across all of my DDA data sets for that peptide being present? And so actually, we'll get to questions about what is good about the different windows as we go forward. But just to answer Leanne's question about the plus 31 and the plus 50 for the DDA data, these are just the number of peptides that were detected in those different regions. So for example, of the 500 peptides that we asked PRM questions for, we were only able to detect about 350 of those peptides using the best DIA methods, and maybe about 330 in those cases for the best DDA methods, in terms of the types of peptides that we felt we could answer, we could quantify. Okay, so I just want to bring up a couple of pitfalls that we should go through before we start to think about collecting this type of data. And so one question I think is this one. So what's going on in this particular case where I've got exactly the same precursor mass? This is just selected out in Skyline. One particular precursor mass for a phospholipeptide. What's going on here? This is the left is the peptide I was actually interested in, but what is this peptide on the right? This is actually just a positional isomer. It has exactly the same precursor mass. So for example, this is a CDK1. It can be phosphorylated at multiple sites in CDK1. Both of these peptides have exactly the same precursor mass. And actually only a very few number of fragment ions. In this particular case, B5 and Y6 are the only fragment ions that actually are different between these. And they have potentially the same or different retention times. So if you start to think about this across time, we'll actually see these as isobaric peptides. These peptides have exactly the same precursor mass. We will see two precursor peaks, but specific fragment ions that indicate one form over the other. And as Lindsay said earlier, with PRM, you need to be careful about monitoring these specific fragment ions. Again, with DIA data, you have to be very concerned that you are using those specific fragment ions to detect those peptides. Because if you're not limiting yourself to using those specific peptides, then you may detect peptides based off of only the shared fragment ions and not necessarily get a trustworthy result here. So here again in Skyline, this is a particular case where at the top I've got a precursor trace where we've got, for this particular peptide, we've got a monoisotopic peak on the left and then no monoisotopic peak on the right, but we see the +1, +2, +3 peptides and exactly the same fragment ions. In this particular case, this is a deamination. So the peptide has been shifted by one Dalton, and as a result, we see a lot of the same fragment ions in many of these cases, but slightly different precursors. And so this is a case where using precursor signal is actually very helpful, but retention time is actually really important in these particular cases because deamidations typically push retention times a little bit further down. A lot of these modified peptides are actually much more complicated than that. Deamidations make sense to be confused because we've made confusion, well, we've confused those before with DDA data here too. But we actually can make very difficult decisions about particular types of peptides where it can be hard to be confident that we're actually making the same types of measurements. So this is an oxidized peptide. So for example, in this particular form, the top one is the unmodified form and the bottom one is the oxidized form. All of those Y ions from Y1 to Y9 are exactly the same. the only thing that's different that's a Y ion is Y10, and there are a bunch of B ions that would potentially be different. But these have different precursors, so they typically wouldn't be confused with DDA, but they may fall in the same precursor isolation window. So if you have a wide enough window, then that can be complicated. And this is particularly an issue with lots of types of these experiments, not just wide window fixed isolation experiments. Even if you use variable width windows, there are regions at the high MZ range where you actually have very wide isolation windows. But if you look at the fragment ion traces for these, so these are again the integrated fragment ions for these DIA experiments, we actually see a lot of the same types of basically all of the same types of fragment ions except for a handful of B ions. Another issue is that homologous peptides often also share a lot of these types of fragment ions too. So for example, this is a particular peptide that I was interested in monitoring in kidneys. This is an actin B peptide. And so there are two different homologous forms of this actin B beta peptide. And if you were to score this from a peptide-centric mode, so for example, in that one on the left, you would actually score it over time where you would see some low scores, and then all of a sudden you see a nice peak, and then you would say, okay, this seems like a really good peptide, it's definitely present. And if you were looking for the peptide on the right, again, you would sort of march along over time, you would see a nice peak, maybe not quite as high, but it actually, it would be a very good peak, and you would select that and say, this peptide is definitely present too. But one of the things that's important is you need to consider these peptides in the context of each other. And this is something that is actually still an open question in DIA. How do you best deal with this type of data? Where in this particular case, only the peptide on the left, the one that has unique sequence ions for this particular form, is actually confident as being present. And this is actually confirmed by GTEx data, where we actually have RNA-seq data indicating that this particular isoform is only present in kidneys, which are the types of samples we were looking at, whereas the other peptide has no signal in kidneys. So again, these fragment ions that are distinctive of positional isomers with phosphorylation, actually we see exactly the same types of issues here in these particular peptides as well. This is actually a complicated case where you've got a D to an E shift, that's a methyl change, and then a V to an I shift. Again, that's another methyl change in the other direction. So these peptides have exactly the same fragment precursor as well. So this would be confusing even for DDA. So I want to talk, before answering some questions, a little bit about setting up a DIA experiment. So I want to refer back to this paper that Lindsay and I just published actually just a week ago in MCP. This is actually, I think, illustrates a lot of the sort of questions that you would ask yourself of why you would, or how you would want to answer setting up a DIA experiment. So I want to refer you to this. There's a link to it in the doc as well. This is, I think, going to cover a lot of this in more detail, but I just kind of want to touch on sort of the highlights here. And one of these things is, so why are we measuring from 400 to 1,000 MZ here? In most of the types of experiments that I've been describing to you, when the original swath paper actually described measuring peptides all the way out to 1,200 or 1,250. And that actually has to do with the distribution of peptides in different samples and in different data sets. So for example, in the pan-human library, we actually can measure most of the peptides out in this 400 to 1,000 range. There are some peptides that we don't see because they're too low or too high, but most of the peptides can be covered in this range. But you might actually want to make a slightly different choice if you were studying phosphopeptides, for example. In this particular case, you might want to shift the masses up a little bit. That said, you're not going to be able to detect all possible peptides in this 400 to 1,000 MZ wide range. So if I were to take acquisitions of narrow window data. So these would be 2 MZ wide isolation windows, where basically we've got multiple acquisitions spanning each of these regions. So for example, 2 MZ wide windows, I can cover that in 4 injections of 500 to 900. Let's use that as a baseline. So if I expand that out to 5 injections, then I get 20% more peptides. Or if I expand that up to 6 injections from 400 to 1,000, I get 30% more peptides. And we start to see, we still get diminishing returns, but we still see an increase in the number of peptides that we see at higher ranges, even though in general, we would sort of expect to see those same peptides at essentially a higher charge state in the lower range. However, if we compare this to wide window data, that's where we are actually limited, where we have a single injection per sample. Because in these cases, we actually need to widen the precursor isolation window range. We actually end up in situations where if we keep the number of scans constant, we actually start to see diminishing returns, and not just diminishing returns at higher MZ, but we actually start to see a drop off as the precursor isolation range widens larger and larger. And so typically we see 400 to 1,000 as being kind of like a nice sweet spot in terms of our detection rate in wide window DIA data. And so that's typically why I would recommend this for most experiments. Again, for phospho experiments, you might want to think about 500 to 1,100 or 500 to 1,000 in those cases. So we have a document that actually describes recommended DIA settings. A bunch of you have asked a bunch of these kind of questions of what you would recommend starting with for a wide variety of instruments. So we have these listed in this Google Doc here, as well as in the manuscript. But there are some additional questions that I think are pretty important to ask about how you set up these types of experiments. So one of the things that we recommend in terms of these schemings for data acquisition are to use staggered windows. And what do I mean by staggered windows? So staggered windows, and there are a bunch of questions in the Q&A. Hopefully this tackles a lot of those. But the idea here is that in every other windowing scan, we would actually take every other set of sequences and then actually tile it over by 50%. And in this particular case, we would actually see information that is sort of like not exactly the same in each different cycle. We actually see it split. But we can use this in some ways to actually be able to leverage it to get more information out of this. And I highly recommend that you use staggered windows going forward. So how does this data actually get analyzed? So for example, in this particular case, I've got a 24 MZ wide window, and all of these fragment ions are listed here in black. But if we start to use previous information from the cycle before or cycle after, some of these fragment ions are going to be present in all 5 of the windows, like this red ion, for example. This one we probably don't know so much about, but there are going to be some ions that are only in the left windows. So in this particular window from 600 to 624, these we are confident are only in the 600 to 612 range. And vice versa, there are going to be some fragment ions that are only in the upper range too. Most of those ions. And then there will be some ions that don't repeat whatsoever. These are frequently noise, or these are typically thought of as noise. So one of the things we can do is sort of extract out in silico these into 2 different spectra that are actually of narrower precursor isolation windows. So essentially we can take our 24 MZ wide window data and generate 2 12 MZ wide scans. We think that this is a really good method for trying to generate narrower scans, at least in Orbitrap style instruments. And that we highly recommend trying to think about this. Oftentimes you'll see, and there was a question about this earlier, about having transmission efficiency of the margins or the areas on the wings of that isolation on the quad. And so the idea here is that you could potentially widen that slightly by adding in little margins on either side typically like half a Dalton or something like that, and then essentially get flat transmission across. This actually is a pretty good method, and we highly recommend doing this if you are not collecting staggered window schemes. But as you might imagine, these margins actually make kind of a mess of how you would do the deconvolution with staggering. And so we typically actually suggest you think about this from a different perspective, which is to take advantage of regions of the proteome where there are no peptides. So there are parts, because peptides are made of the same materials, it's all hydrogen, carbon, nitrogen, oxygen, sulfur, there are only so many spaces in the precursor isolation space where you can actually make a peptide exist. And because of that, there are these boundaries that exist where you could say, well, I'm not going to see a peptide here, plus or minus 0.25. And then because of that, I can be confident that if I set my window boundary in that region, it's not going to have an effect, or my isolation window efficiency isn't actually going to be an important issue with these particular cases. So we actually really recommend trying to think about this as a strategy rather than increasing the window widths with margins, at least if your transmission efficiency is pretty good, as is the case with most modern quadrupoles. That said, these ideal dead zones actually depend or change depending on the type of data set that you're looking at. So for example, if I take all of the possible peptides that are in Swissprot, all the +2s and +3s, that's that trace in green here. And if I look at the peptides that are in the pan-human library, some of those are modified peptides or PTMs, they're acetylated or oxidized peptides. There are some slight variations off of the default Swissprot, but it mostly lines up pretty closely. But if I look at that phospho library that I was looking at before, this actually introduces a slightly different mass defect, and you actually need to use slightly different dead zones. So we've got a method to do this calculation in the paper, as well as a software tool that will do this too. So I'm going to answer a couple of questions before getting into this last section about building libraries. And just so that we can make it through everything, I will answer a couple of these questions, but then I'll come back to them at the end for anyone that wants to stick around for as we're going forward. Okay. So again, going down to the bottom, so the last question is, do we need to collect data in centroid or profile mode at the fragment ion level? There's a lot of debate about that, actually. Typically, I collect all of my data in centroid, even for precursors at this point, because the data files are just so large that it's much more convenient to work with them as centroids. I think this is particularly beneficial for thermo instruments where essentially you're doing centroiding anyway because you're going through the FFT process, as opposed to writing in sort of essentially fake profiles in those particular types of instruments. That said, there might be some reason to use profiles in certain types of experiments, particularly where you want the most sort of sensitive data analysis, where you might have a specific peptide that you're interested in specifically that you can only see at a very low level, and that won't actually get picked up by the centroiding algorithm. So that is definitely a potential issue. But I personally collect all of my data in centroid mode, again, for both fragment ions and precursors. So if you're making decoys, and this is a really great question, if you're making decoys with the end amino acids are the same, then those B ions and Y ions on the ends are going to be exactly the same. And that is exactly true. And that's actually sort of the point, is because, for example, if you took that arginine or lysine on the N terminus, almost every peptide in our data set produces arginine and lysine. So if you look for Y1 in a DIA experiment, you will see it constantly all the way across the entire data set, where there's the peptide that you're interested in and all other possible peptides, because every other peptide has that same fragment ion. And so it's necessary to make sure that you are decoys have that same characteristic, because if you actually can move, for example, Y1, then it's trivial to tell whether a peptide is a decoy or not, because it won't have that same Y1 signature. So that's why, one of the reasons why it's really important to keep the same kind of characteristics of the termini. Again, the same thing happens with B1 too. It's just 1 out of 20 amino acids in that particular case that can fall on that N terminus. As you start to narrow your way in, those frequencies get less and less. And so typically we don't like to extract a lot of data that's B2 or Y2 as being significant. And typically we start to count after Y3, unless you need a specific ion to call that peptide. So for example, if you have a phosphopeptide and that's the only ion that can help you with. So, so if you change the window size, you actually can change the sort of complexity of the experiment. So for example, 10 MZ wide windows are definitely going to get less complex spectra or MS2s than 25 MZ wide windows. That's definitely true. But you have to counter that with either sacrificing number of points across the peak or the total window width that you're interested in measuring. That's one of the reasons why we think that the staggering methods are actually a bit better than actually having lots of different narrower windows. So for example, that 24 MZ wide window that we were talking about, essentially equivalent to your 25 MZ wide window, we can cut that into 2 12 MZ wide windows using staggering for free, essentially. And because of that, we actually get a pretty significant benefit. On faster instruments where we can scan faster, we actually recommend even cutting down further. So for example, on a Lumos, we might use 16 MZ wide windows or 8 MZ wide windows and actually have staggered those so that we can get the equivalent of 4 MZ wide isolation windows after deconvolution. Okay, great. I'm going to answer these other questions going forward, but I want to talk briefly about why it is difficult to use DDA libraries in some cases, and why you might want to consider collecting DIA-based libraries, and how we can best deal with that. And one of the answers for that is using peptide predictions like Prosit, for example. Okay, so in terms of DDA libraries, if you're going to do a massive amount of fractionation, it can be a lot of work. So for example, the pan-human library actually used over 300 injections to generate. Our phosphopeptide library had over 1,000 injections to cover the human phosphoproteome. So a huge amount of data generated from these types of measurements. And one of these sort of reasons why, or one of these sort of issues, is that how you generate your fractions actually is really important. So for example, if you compare the peptides you can detect, again, triptych peptides, normal triptych peptides you can detect using SCX fractionation, you actually get a very different profile than the peptides that you get with high pH reverse phase fractionation, which really means that if you want to generate a full library, you kind of need to do both at the same time. And so this doubles the amount of work that you need to do. Potentially, if you use other types of fractionation methods, you would again have a different level of overlap. And so there's a lot of data that you end up needing to typically acquire if you're generating DDA libraries on your own. The other aspect is that DDA libraries aren't necessarily very accurate. So for example, here I've got a spectrum on the left for a particular peptide where I've got a DDA spectrum for it and a DIA spectrum that I've acquired. And so this is a +3 peptide that was acquired in DIA mode, where the collision energy isn't necessarily perfectly optimized for that specific peptide. So here, because you need to select a specific charge state to be able to optimize the majority of your peptides for, In this particular case, I had chosen +2 as the strategy. Typically these days, I actually recommend you use +3 instead. But because of these sort of characteristics, the fragmentation patterns aren't necessarily representative of the exact fragmentation patterns you should expect to see in DIA versus DDA. So for example, here I've got on the bottom of that left panel, Pearson's correlation coefficients for these particular peptides. In this particular case, the +2s look pretty good. The +3s are good too, but the +4s are kind of garbage in terms of their actual accuracy. Again, because the instrument was tuned to fragment all peptides as if there were +2 peptides. The other aspect of this is retention time isn't necessarily accurate either. So for example, if I use retention time from a DDA library where I've done fractionation, it actually has that peptide I'm interested in monitoring in a background of a very simplified matrix. And so how it elutes off the column actually changes a little bit depending on what's around it. And because of that, we actually see these kind of curious banding pictures when we actually measure it in the full matrix with DIA. So we actually see some of these characteristics where the true fragment retention times are slightly different than the actual retention times, and it's really hard to nail down that precisely, or at least within a minute or two. These are typically 5-minute windows, so we would need to do over a 90-minute gradient. Okay, so this is a slide that Mike McCoss likes to show, which is that we typically talk about DIA as a digital archive of an entire proteome. And so this is a slide that shows a digital archive of the Trump inaugural election or the Trump inauguration that happened in 2016 that Google took. This particular picture lets you zoom into this megapixel picture, lets you zoom into individual people. So for example, here's a picture of President Obama looking pretty tired and potentially a little sad. But realistically, the DIA data that we collect doesn't really look like this. It's not got the same type of resolution. It's not got the same type of accuracy or fidelity that we have to say. It is comprehensive, but it is not necessarily precise. So we end up with these really pixelated pictures. And then when we take a spectrum library that was acquired of a different Obama with a different tie, for example, and maybe less gray hair, it might be actually pretty difficult to try to match our spectrum library match to the actual, our spectrum library Obama to the Obama that was found in our particular sample of interest, this inauguration Obama. So one way to sort of help with this is to use gas phase fractionation. So gas phase fractionation, I think, is a really clever idea developed by the Goodlet lab originally about 10 years ago, over 10 years ago, where they actually use multiple injections for a single sample. So they'll take that same sample, and instead of measuring from 400 to 1,000 MZ with 24 MZ wide windows, they'd acquire it in 2 injections, one at 400 to 700 with 12 MZ wide windows, or 700 to 1,000 in a second injection, again with half-size windows. This is a very powerful method to be able to get much more accurate or highly resolved data. We actually have been starting to explore how to do this at a grander scale. So instead of 2 injections, why not 6 injections? So where we actually monitor from 400 to 500 in 4 MZ wide windows, and then 500 to 600 and 600 to 700. Now, if we stagger these 4 MZ wide windows, we can effectively get 2 MZ wide isolation windows, which is essentially the same quality as PRM data, except instead of PRM for a handful of peptides, it's PRM for all peptides between 400 to 1,000. So it's an extremely rich type of data where it's easy to detect peptides, it's easy to monitor peptides. So this would be way too expensive from an instrumentation perspective to do for every one of our quantitative samples. But we've started to do this for generating libraries. And this is demonstrated in a paper that we put out a couple of years ago, where we would take n quantitative samples, where we would do single injection DIA on each one of those individually. So that would be say if we had 50 samples, 50 quantitative samples, we would have 50 of these 400 to 1,000 injections. But we would also we would skim a little bit off the top of each one of those. Either we would pool it beforehand, or we would take a microliter out of each well afterwards. We would generate this pool, and then we would analyze that pool using the 6 gas phase fractionation approach. We would search that using a spectrum library or even a FASTA database search engine strategy, and then use that to build what we call on-column chromatogram libraries in exactly the same matrix of exactly that same sample on that same instrumentation platform. So this is as close to the original type of acquisition as we could ever get, and give us a chance to be able to make these detections in as easy a way as possible in the 6 gas phase fractionation strategy, and then sort of transfer those detections over to the end quantitative samples. So in a sense, that's sort of like taking this DDA spectrum library approach, but breaking it up into multiple different precursor isolation windows. So 400, 1,000, 600, 500 to 600, 600 to 700. And then zooming into that place with Obama here, for example, where we actually can collect PRM quality data and then zoom into that. It's much easier for us to be able to associate the high quality measurement of Obama with our spectrum library or FASTA database search, and then now equate our wide window data with this narrow window DIA data on the same instrumentation platform with the same level of specificity. So let me show you what this actually means. Here's the retention time accuracy versus a DDA library. This would be the equivalent retention time accuracy using one of these chromatogram libraries. So instead of being plus or minus 5-minute windows, we're actually plus or minus 20 seconds in these cases. And the same thing goes with fragmentation patterns, where all of the spectra, all of the charge states actually show very good representation of correlation from one sample to the other. So we see typically very high improvement in terms of the number of detections that we make between single injection measurements. So for example, we can essentially, when considering a library, be able to see about 50% more peptides than we would typically see with that same type of experiment, just using a DDA library. I want to say something about when we would acquire these samples. And the place at which we would typically want to acquire these, because we need to break in a column over time, we typically want to acquire these after about 5 or 6 or 10 different replicates. So typically we would acquire one replicate analysis here, acquire our pooled library, and then our other samples in terms of our acquisition order. So this way we can ensure that the library doesn't need retention time alignment between each of the gas phase fractionation samples. I want to get to, and this is just very quickly, this idea about being able to use Prosit predictions or predictions to generate these libraries. One of the problems, as we said, or the biggest advantage of a spectrum library actually isn't that the fragmentation patterns are good. It's actually the narrowing down of the size of the library. So fragmentation patterns get you something, but being able to work with 10,000 peptides instead of 100,000 or a million peptides is a huge improvement in terms of your specificity. And so one of the reasons why it's very difficult to just predict all peptides in a sample or in a proteome is because of this sort of issue, where if you were to predict all of the possible peptides, you still have to search all of those possible peptides. We've actually been applying the chromatogram library strategy with these Prosit libraries. We'll actually take a FASTA database, we'll predict all of these possible peptides, and then search the gas phase fractionated samples in these particular cases, and generate with these 6 runs and these predictions actually very accurate gas phase fractionated libraries without having any spectrum library at all, because it is so much easier to detect peptides in these gas phase fractionated samples, these PRM quality data sets. And again, using that same framework, we would actually collect our single injection wide window DIA of our biological samples, and then line those guys up, and then have very highly accurate data from one sample to the next in terms of our library versus our single injection data. So I showed this slide earlier, but we can essentially get the same quality of detection using the chromatogram library approach based off of a spectrum library as versus the predicted library. So using the gas phase fractionated data sets to kind of consolidate and fix the sort of predictions and narrow down the number of spectra that we're actually doing in our comparison, we actually essentially can get as high quality on column data as possible with this type of approach. And so we highly recommend trying to use this strategy going forward rather than trying to develop these large spectrum libraries. And so we've built a workflow here to be able to do that in Encyclopedia, where we have this nice graphical step to be able to generate these libraries, which we have laid out in this paper that was published a couple of months ago. So with that, I'd just like to conclude that, again, DIA is an excellent strategy, but it's all about compromises in these particular cases and making the right choices for what types of compromises you're willing to take. That we talked quite a bit about analyzing DIA data and possible pitfalls of interpreting DIA data, specifically around post-translational modified peptides, as well as sequence similar peptides. And then finally, we talked a bit about how you would best set up a DIA experiment. And then finally, building these libraries for DIA experiments. So with that, I would like to acknowledge several people here, especially Lindsay and Mike, who have contributed a huge amount to this type of work, including a lot of this new sort of work. And again, I want to suggest referring back to that paper that is at MCP that was just published. So it's easy to, it's freely available to access at this point for the next 3 weeks, I believe. So you can get that even if you don't have an MCP subscription. So with that, I'll, I guess, hand it back over to Olga, and then I guess start to answer some of these questions as we go through.
Thank you, Brian. I think we can take like some question to take an answer, like, bunch of, tons of the, questions are waiting for you. Maybe just 5 minutes. And then we can post a copy all the question in the Google Doc. And then if we have a chance later, you can answer the question over here.
Yes, perfect.
Yeah.
Yes, so I'm definitely happy to do that, to answer questions in the Google Doc. Or if you have additional questions, feel free to either tweet at me or email me or stick those straight onto the Google Doc as well. So for these 5-minute questions, I will start again at the bottom and then go up. So how accurate are DIA peptide detections if we don't actually know anything about the peptide precursor? And how confident are we in these types of measurements? So that's actually an excellent question. So oftentimes we actually see situations where we acquire peptides where with DIA that we can make really clean detections of them, but we actually don't see a precursor for those peptides. And these actually are situations that are caused due to this phenomenon where you can only spend so much time and ions filling an orbit trap or collecting a precursor scanner in a TOF. So thinking about this from the standpoint of an orbit trap, if an orbit trap can only hold 1 million charges and you've got a really high peak, say for example you're working in serum and you've got an albumin peak that is at 1,000 MZ range, it's going to be really hard to see a precursor in that same window because 99% of the ions of those million ions are going to be that one precursor peak. So oftentimes we end up being able to see no precursor signal because there's a huge other ion present at that same time point in the precursors. But in the fragment ion scans, we actually see really good fragment ion peaks because we are selecting out only fragment ions that are associated with this narrow precursor peak in the range that we're interested in, 500 to 525 or something like that. Again, this is one of the reasons why accurate time and mass tag methods, like how the Matthias Mann group has been doing these, and Dick Smith's group did these, 15 years ago, where you would actually have very narrow precursor isolation windows, where you were just collecting very high quality precursor scans. This is the reason why those types of methods work. But from our perspective, we figure why collect just a precursor scan when you could just be collecting fragment scans instead, because the fragment ions are much more specific, and you also have the ability to throw out fragment ions, as opposed to with a precursor where you only have those 3 measurements at your fingertips. Okay. So how accurate is Prosit? Because it was built for orbit traps. And is it accurate for other types of orbit traps? Because it was designed around lumoses, for example. And then also, does it work for other types of QTOF instruments? So actually, it again, as I said, it's designed, Prosit is designed for lumoses. So if you're using a Q exactive, as I said earlier, you should take 6 off the NCE and use the reported NCEs as if there were 6 above. So for example, if you acquire data with an NCE of 27, you should tell Prosit you acquired that data, or you are interested in collecting spectra at an NCE of 33 instead. So that's the easiest way to get as close to equivalent spectra as possible. That said, these predictions actually, because they're both beam type methods, using HCD, you actually get very good close fragmentation to what you would typically get in another type of beam type instrument like a QTOF. That said, if you were collecting in CID mode in a orbit trap, you would actually not get very good spectrum quality in the same kind of way, or comparisons in the same kind of way. So the Prosit predictions actually work really well for the QTOF. That said, they are accurate only to a degree. They're good, but they're not perfect. And so that's one of the reasons why we developed this approach to use gas phase fractionation to essentially correct the Prosit predictions, both in terms of fragmentation patterns as well as retention time standards. Okay, so do we need to use IRT standards in our library-free methods? Actually, no, there's no real reason to use IRT standards in these cases, because essentially using our approach, using the gas phase fractionation, we use all of the peptides as IRTs essentially. So we do a fitting first without any knowledge of retention time, using these essentially retention time free strategies. And then we use retention time as a correction after the fact to choose different sorts of peptides in those particular cases. Okay.
Just one last question.
Great. So can encyclopedia work for MSE data from waters without ion mobility data? In that particular case, I think that's a very difficult type of data set without ion mobility. And there, I think your best bet is to actually still use Waters software. In that particular case, Protein Links Global Server as the sort of best way to interpret that type of data. I wouldn't necessarily use encyclopedia in those particular cases. However, if you did gas phase fractionation, or if you did narrower precursor isolation windows, multiple narrow precursor isolation windows, you can still use a Waters QTOF and analyze that data with encyclopedia, as long as you do a more sort of traditional DIA type of experiment. Okay, excellent. That seems like great. I will respond to the rest of the questions in the Google Doc afterwards. Thanks.
Perfect. Thank you so much, Brian, for a great presentation. And then also thank you for participants for your interest. We record all the presentation. It will be available in YouTube in few hours. We just need some hours to compiling the files. So all the links also will be available in the May Institute website. If you sign up, see you tomorrow. Thank you again. Bye.